talking about natural herbs as a potential treatment option for neuropsychological impediment and diabetes and Alzheimer's disease. Dr. Saara.
Very much. Thank you very much for introducing me.
Yeah.
Can I share the slides from my side, or you're going to share my slides?
You can do it if you want.
Okay.
Okay. Thank you.
Right. So you can see the slides.
Yeah, sure.
So I'm Dr. Saara Ahmad Muddasir Khan. I'm assistant professor at Biological and Biomedical Sciences, Ahmad University, Karachi, Pakistan. So my topic is natural herbs as potential treatment option for neuropsychological impediments in diabetes and Alzheimer's disease. So Alzheimer's is also known as type 3 diabetes mellitus. So what is diabetes mellitus? Diabetes mellitus is a metabolic and endocrine disorder where there is hyperglycemia due to decreased insulin production or increased insulin resistance. So the either insulin is not working or it's not there. This hyperglycemia leads to oxidative stress, and the oxidative stress eventually causes the neuroinflammation. The inflammatory response to the oxidative stress causes the neurons to get damaged, while the inflammation of the mitochondrial membranes and the cell membrane. So once the mitochondrial membranes are damaged, we know that most of the monoamine oxidase enzymes, etc., they are present in the mitochondrial membranes, and these are all secreted out. Once they are secreted, they causes the imbalance of the neurotransmitters, especially serotonin, dopamine, and norepinephrine. Along with this, it also causes the apoptosis of the neurons. And besides this, the neuroinflammation or the inflammatory processes with the imbalance of the action of superoxide dismutase enzymes, catalase enzyme, and then the fatty acid metabolism and the fatty acid oxidation leading to the formation of malondialdehyde. It causes the induction of A-beta protein productions and tau pathology. So all these things come into play and lead to the Alzheimer's disease. For all these reasons, Alzheimer's is also known as type 3 diabetes mellitus. It is also seen that those patients who are diabetic, they are more prone towards getting the Alzheimer's disease. And hence, to control diabetes and to control the blood sugar level is the best way to control Alzheimer's neuropathy. Now, there are many FDA-approved drugs available in the market. These FDA drugs are not only costly, but have side effects. Besides these side effects, there is no permanent cure to Alzheimer's disease. There's no permanent cure to all these neuropsychotic disorders. So now there's a trend towards opting the natural function foods for the treatment of these disorders. WHO has also recommended the use of natural products for the treatment of diabetes and its allied disorders. Now, there are many anti-diabetic natural herbs which have possessed the properties of controlling blood sugar levels, which have the property of controlling those oxidative stresses, along with the inhibition of the tau tangle formation and amyloid beta protein synthesis. Now, among these anti-diabetic natural herbs, we have chosen saffron and chamomile. Saffron is a costly herb, while chamomile is not that much costly. They are easily available in Pakistan. They are easily available worldwide. Even chamomile tea can be found anywhere in the market. Dilma company and so many other companies, it's like they're available on the shelves. And saffron is also available on the shelves of the supermarkets. So these two are commonly accessible, and they have the ingredients or the phytochemicals which are shown to have beneficial effects against treatment of diabetes and Alzheimer's diseases. So we have taken these two herbs to to evaluate their efficacy against diabetes mellitus and Alzheimer's disease. But the novelty of this study is this, that these herbs are not given only separately, but also in combination to the animals. Now, the experimental model was done in such, the experimental study was done in such a way that we took the animal model. The animals were made diabetic at first, and then they were left diabetic for a certain period of time so that they should start having the neuropsychological disorders. And once they have the psychological disorder, we did the neurological behavioral test. And after the behavioral test, these rats were then given all these treatments, in which they were given the water decoction of chamomile, water decoction of saffron, and water decoction of combined co-administered saffron chamomile, but the doses were reduced. The doses were reduced to half. These were given for 2 weeks, and again their cognitive abilities were tested and their behavioral tests were done, and after that they were sacrificed. The blood was calculated, their brains were preserved, and estimation of serum tryptophan was done in the blood, as well as serotonin and the other monoamine neurotransmitters in the brain, while the behavioral tests were done for depression and for memory. The background of doing this tryptophan analysis was this, that the tryptophan is a precursor of serotonin. So this tryptophan, it has to go to the brain, cross the blood-brain barrier, enter into the brain to form serotonin there. Serotonin is formed in all parts of the body. GIT is the heaven for serotonin synthesis. But this serotonin, which is produced anywhere in the body, cannot cross the blood-brain barrier. Since it cannot cross the blood-brain barrier, it is not beneficial for the brain. So the brain has to prepare its own serotonin. Now, the test which I am going to describe here is the novel object recognition test. This novel object recognition test is this, where the rat is placed in a box with two identical substances, and it's allowed to sniff them. It's allowed to see, sniff, and recognize these two objects. So this is a habituation or the learning phase, and after that, their memory is tested. So short-term memory is tested and long-term memory is tested. After 2 hours, again the rat is placed in the same box. One object is the same while the other object is a different object. As the rat has already sniffed the object, so the object that was previously present is going to spend less time if it does not have any cognitive disorder. It is going to spend more time with the new object, that is the novel object, because that's new in the box. So after 2 hours, if the rat is showing interest in the new object, it means that it has maintained a short-term memory, and it knew that the other object, the older object which was already in the box, it has previously sniffed it, and now there's a new object there. But if a rat has a cognitive impairment, it is going to spend more time with both of them. Like it will spend more time with already placed or already sniffed object and not the novel one, or could be equal time with both of them. And then the long-term memory is done after 24 hours. Again, one object is the same, which was previously given during the habituation. And then again, another novel object is placed in the place of the other this object. Again, the sniffing time is tested. And then if the rat is spending less time with the older object, it means that it has maintained its cognitive abilities. However, if it is not doing this the way it is expected, then it means that there's a cognitive impairment. Now this is the brain. This is the brain of a rat. We excised the brain from the skulls, and the brain were sectioned. Cortex, hypothalamus, and other parts of the brain were separated, and the tryptophan levels were done in the whole brain. So for the hypothalamus and for the cortexes, they were all being utilized for different other analysis for receptors, etc. Well, the whole other part of the brain was done for the tryptophan levels to see whether the tryptophan was able to cross the blood-brain barrier. Because in diabetes, the flow or the passage of the tryptophan through the blood-brain barrier is hampered. One, the insulin helps its mobility. So once the insulin is not there or the insulin is resistant, then again tryptophan would not go into the brain. So the whole brain was estimated and HPLC was done for the estimation of tryptophan analysis. The neuronal integrity was done and was seen by the histology. In the present study, we saw that the hubs, they very clearly control diabetes because animals were routinely and daily tested for in the fasting blood sugar level for their glucose levels, and it was seen that the herbs individually as well as in combination controlled their diabetes and cognition, improved their cognition, which were tested in the novel object recognition test. Now, one thing is worth mentioning here is that the animals with the group of the animals that was given the combination of the herbs showed more improvement in cognition and more control on the fasting blood sugar than compared to the rats which were given individual herbs. Tryptophan analysis also showed the same results, that the group which were given the combined decoction and half doses showed higher tryptophan level in the brain, and the brain slides showed the improved integrity of the neuron. There was less vacuolization and the less tau protein production and the tangles formation. So in the rats, there were two different groups of the rats, and again, in animal model, Alzheimer's is different than the human model because the human presentation, because the humans, they can have both tau and both amyloid beta plaques, but in animals either we are going to have tau or we are going to have the amyloid beta. So in our study, the histological slides showed that there was tau tangles production. So the animals that were given the herbs had the better brain morphology, but the best was those diabetic rats which were treated with the combined half decoction of saffron and chamomile. Then this present study demonstrates the potential and cost effectiveness of the traditional treatment for diabetes and Alzheimer's disease and its subsequent complications. Now we see that in our part of the world, where India, Pakistan, Bangladesh, we have a strong belief in the natural products, and people, they are more prone and they show more acceptance towards the natural products than towards the allopathic medication. And it was seen that yes, these herbs can have a beneficial effect. In the process, this study is in a translational phase in humans, and we are going to see in humans what is their effects and how it's going to affect their other behavioral disorders, which include anxiety, depression, schizophrenia, Alzheimer's. So the human translation study is underway, and the animal study has shown that the things are getting better. So so the other thing that I want to explain here, or to say here, is this, that the decoctions are a very easy process. It doesn't need any sort of a specialized instrument or any sort of a specialized procedure. Just the herbs are going to be taken or weighed according to what is supposed to be given to an individual, placed in the water, start simmering. When it comes to boil, let it simmer for 20 minutes, and then give it to the person. So this is the decoction. Any lay person can make it, and it's very easy. So these herbs showed the effectiveness in the treatment of diabetes and Alzheimer's diseases, and I hope that they are going to feature more and more of such herbs, and studies will be on the way to show that they are beneficial and have the effects. And this is not actually, if I touch the background now, it's not actually new, because metformin, the miraculous drug which is used nowadays, for diabetes and so many other disorders has already been taken out from a plant. It's a plant extract from the Galligia officinalis plant. So it's not new of having these things, but then again, these herbs, once they are tested and they show positive, then we can go for the deconstruction of their elements and their phytochemicals and see that which chemical is more effective against the treatment of diabetes and Alzheimer's. Now, so thank you very much. It was the crisp and short description of what we did. I appear as Saara Ahmad with my publications, and you can see my publication on PubMed and the Google Scholar, and the links are given in it. I thank you very much for listening to me, and I am